Hi, we're Group 2 and we're presenting an overview of the molecular mechanisms for cell polarization during early embryo development as proposed by a recently published paper. Cell polarity is crucial during embryogenesis because polarization of cellular material aids differentiation. This occurs during the 8-cell stage, which is 2 to 3 days post-fertilization. During the stage, the apical and basolateral domains are formed. This crucial step directs the bifurcation of the trophlectoderm and the inner cell mass, specified as TE and ICM in this diagram. If this is defective, developmental arrest can occur. Partitioning defective proteins or PAR proteins are integral to this process, and Rho proteins aid in maintaining this polarity. Although cell polarization is crucial, the exact mechanism that initiates it remain unknown. We're going to focus this presentation on the results of this paper that describe the role of phospholipase C. Specifically, they propose that PLC-mediated PIP2 hydrolysis is the initiating event for cell polarization in mouse embryo development. This paper first looks at the localization of proteins that are essential for cell polarization in the 8-cell stage of embryogenesis. Figure 1a on the left shows fixed 8-cell mouse embryos at the early, mid, and late stages respectively. F-actin and PAR6 are immunostained with green and purple markers respectively. F-actin acts as a marker for actomyosin and PAR6 acts as a marker for the PAR complex. In the early phase, actin is polarized to the apical domain and is important for recruiting PAR6 and forming the mature apical cap. In 1c, we can see as compaction of the blastomere increases, there is movement of both actin and PAR6, whereas in phase 1, in the initiation phase, only actin, the green line, has migrated. The movement of F-actin and PAR6 during the development of the embryo provides evidence that the cell polarization does occur in a stepwise manner, which is summarized in the cartoon shown in 1D. This paper demonstrates that PLC-PKC signaling is necessary and sufficient for triggering the actomyosin polarization we mentioned earlier. To test if PKC is required for compaction and polarization in the embryo, researchers applied potent PKC inhibitors, sphingosine and calphostency, as indicated by the schematic in Part A, to mouse embryos at the 4 to 8 cell transition state. When immunostained for F-actin and PAR6, the DMSO controls showed significantly more fluorescence. Both treatment conditions limited F-actin and PAR6 accumulation and averted compaction. This data was also succinctly presented in the form of a bar graph, indicating that there were significantly fewer cells undergoing polarization when treated with sphingosine and calphostency. Therefore, PKC is important for cell polarization. It's previously known that Rho proteins are a class of GTPases that are involved in 8-cell polarization. Therefore, the authors of this paper hypothesized that Rho A might be affected downstream of PLC-mediated PKC activation, through which phosphorylation of actomyosin is controlled. After inhibiting Rho A, decreased localization of actomyosin was observed in the apical domains at the 8-cell stage, which suggests that Rho A does indeed regulate actomyosin localization. Furthermore, using FRET, it was determined that Rho A is indeed a downstream effector of PLC-PKC-mediated signaling. In the DMSO control group to the right, there is a high-intensity FRET ratio at the apical domains, contrasted with low FRET ratio in the U73122 treated sample. This sample contains a PLC inhibitor blocking signal transfer to Rho A. The scatter plot to the right also corroborates this conclusion, as the amount of Rho A FRET does increase with cell compaction, whereas the U73122 treatment abolishes polarization. This paper concludes that actomyosin localization is foundational for PAR complex localization in the apex. PLC-PKC signaling was needed for actomyosin localization through Rho-mediated recruitment of myosin. Initially, adherence in cell contact was thought to trigger polarization, but this was determined to be insufficient as compact 4 cells did not undergo polarization. There are additional factors that do need to be identified that allow for PAR complex accumulation and initiate cell asymmetry, but this paper did demonstrate that polarization and compaction do go hand in hand.